this video we will continue with our introduction to networks and look at more types of graphs like hypercube graphs and so on, and we will also start looking at important graphs in biology, namely metabolic networks, and we will have a brief aside where we discuss about storing and representing sparse matrices on a computer. So, your graphs could be dense or sparse, right, and you will typically find that most biological networks are quite sparse, in the sense there are very few interactions for every node. Relatively, if you have thousands of nodes, then a node might actually interact only with 100 nodes at max, usually with 3 or 4, but occasionally with 100, but never more than 10% of the nodes or something like that. And this has implications for how you do the math with these kinds of networks and so on, right. And then you have cyclic graphs, right. You can imagine that metabolic networks will be cyclic, right. You have several cycles like the tricarboxylic acid cycle and so on. So, you will see that there is a path from a node to itself. So, you start at A on this graph, you can go to on this graph you go from A, you go to C, you go to B, you can come back to A, whereas in this graph you find that there is no such path from A to itself, right. So, trees are essentially connected acyclic undirected graphs. They are again, they can also occur in metabolism. Whenever you remove cycles, you will find that there are directed acyclic graphs that arise, and they also arise in scheduling and so on. Let us not worry about that. Another useful concept is the concept of labeling a graph, right. So, if a graph is unlabeled versus labeled, right. These two graphs are the same, right. If I did not label them, if I label them, the graphs are no longer isomorphic, right. So, till you label these graphs, they were equal, right. So, these have very interesting applications in aligning proteins, aligning protein interaction networks themselves and so on. And then you have this notion of an implicit graph, right. So, these are graphs where you can describe what node connects to what node, but you never spell out the entire structure of the graph or something like that. So, a classic example of an implicit graph would be something like a hypercube graph, where basically this is a 3-dimensional hypercube graph, right. How many nodes are there? Basically 0 0 0 0 0 1 1, and in a d-dimensional hypercube you will have d nodes, 2 to the d nodes, right, and d dimensions, and you will have edges between I and j if they vary in exactly one dimension, right. So, you may never be able to store this graph on a computer, right. If d is 30, 2 to the 30 is a very large number, right, it is like a billion roughly, right. So, you will not be able to store it on a computer, but you can run several graph algorithms, right. And in fact, hypercube graphs have implications for what we will study at the very end of this course. The advanced topics that I mentioned earlier on. So, hypercube kind of graphs are very prevalent there. Another very useful concept is that of bipartite graphs, right. And so, this brings us back to how do you represent metabolic networks. How do you represent a metabolic network? So, this is your network. Right. How would you represent this as a graph? There are various ways to do it, and depending upon your choice you will end up with a different kind of representation. So, one way to represent this would be something known as a substrate graph. As the name suggests, in a substrate graph the nodes are all substrates. In this case it will be A, B, C, D, E, F, and the edges represent those substrates which can be converted to one another. So, A, B, D, do you see a limitation with this kind of a representation? You do not know, right. So, this again brings us to a limitation of modeling, right. So, how would you choose a representation? Going back to how would you choose a model? Depending on what you want. So, if I want to figure out what is the shortest path to produce F from A, or let us just for a moment think about glycolysis. I will just write two reactions, glucose plus ATP is that right? That would be the first reaction of glycolysis. What is the last reaction of glycolysis? Phosphoenol pyruvate plus I have a problem here. I now have a two-step glycolysis. Convert glucose to ADP, convert ADP to pyruvate. Wow, I have glycolysis in two steps. This does not make any thermodynamic sense, this does not make any biological sense, but it is potentially a conclusion you would draw from the substrate graph. One way to get around this is just get rid of ATP, right, because you will find that these nodes have very high degree in the graph, you just get rid of this, then you are stuck. Then you have to find a way to convert glucose to PEP and PEP to pyruvate. You will find that this will help you trace out something that is most likely your real glycolysis. But naturally you want to look for other ways to represent these graphs. Another way to represent this is what is known as a reaction graph. As the name suggests, your nodes are R1, R2, R3. When do you connect them? If they can operate consecutively, right. So, after R1 you can have R2 and R2 you can have R3, right. There is a notion of direction again, but again you see very limited in the information, right. So, since we are not happy with either of these, let us try to look at how a textbook picture would normally look. How would your textbook picture look? It will be basically something like A, you will not have R1 in the box, you would actually have the first enzyme in the box, right, and then you will have B, C, R2 gives D, and D, R3, E, F, right. Do you notice something peculiar about this graph? You will note that there are no links between circles or no links between squares, right. So, you find that all links are between circles and rectangles or rectangles and circles. So, this is called a bipartite graph, because you can partition the nodes into two such that only one bunch of nodes connect with the other bunch of nodes, no connection between the nodes. This also, as an aside, quick aside, how do you represent these graphs? You finally need to represent them on a computer. We will look at it obviously much more closely in the next lab session, where we will have an intro to MATLAB, but we usually use something known as an adjacency matrix. What is an adjacency matrix? It is a matrix size of number of vertices in the graph, Aij equal to 1 if there is an edge between I and j. Immediately you can think of multiple things. For a weighted graph, the adjacency matrix will carry numbers, weights. In an unweighted graph, it will just be binary 0 and 1. In a directed graph, the adjacency matrix will be asymmetric, whereas it will be symmetric in an undirected graph, or you will essentially only store the upper triangular matrix or something like that. How will it look in a bipartite graph? If I said these are my metabolites and these are my reactions, your circles and your rectangles, how will the adjacency matrix look like? They will be non-block diagonal, right? This will be 0, this will be 0, no intra partition links, right? You will have ones only somewhere here, right? So, you will have it will be a blocked matrix. Obviously, this is after you reorder the rows. If you interleave the reactions and reaction and metabolite nodes, you would not have this, but if you reorder them by the partitions, like the R partition and the M partition, you would observe this kind of a matrix, right. And one more important thing you need to think about is how to store the matrix, right. You typically use an array. What is the problem with an array? It takes up or if you are not familiar with big O notation, it basically takes approximately n squared space. So, in fact, if you are looking at storing doubles, let us say how much space is taken up by a double in computer memory? 16, 32, 64, right. So, it depends really on the precision, right. So, most machines will have at least 32 bits, right, or 4 bytes. This is in fact for a float. For a double, it is usually 64 bits and 8 bytes, right. So, you are going to store 8 bytes. So, 8 n square bytes is what you need. But what did we say already? We said that most real graphs or most biological graphs are actually sparse, right. So, which means maybe I do not have let us say the number of non-zeros I have is some alpha n square, where alpha is much less than 1, right. Maybe I have 3% non-zero entries, right. So, it is wasteful to store the entire matrix in memory. I mean, as you know, a full matrix with basically having 97% entries are just being filled up with zeros. So, is there a better way to store these matrices? Is there a better
The non-zero components in the location.
You can just store that would be a classic way. You just store I, j, aij, How much space does this take? Okay, into 8, so that will be 24 alpha n square, and given alpha is we said 0.03, right. So, it will be 0.72, right. So, that is a much smaller number compared to this. So, in practice you will obtain a lot of savings when you store it like this, but this is also a naive method. 0.72 is much smaller than 8, right. So, you have made heavy savings. There are other methods which I want you to go back and read about. There is something known as CSR. It stands for compressed sparse row format. There are many other formats to store these matrices, and the advantage is how fast is it to compute A transpose, how fast is it to compute A transpose X. These are things you would normally learn in a numerical methods course, right. So, how sparsity can be exploited in computations.
CSR is harder than square.
CSR is see, none of this is so n square is actually misnomer here. You should just say it is linear in the number of non-zeros, right. So, this is already linear in the number of non-zeros, this will be again linear in the number of non-zeros, but even smaller. You only store the runs, basically where do my zeros non-zeros start and how long is my run of non-zeros and what are the positions that they occupy and so on. Okay. So, these are some very important concepts, because when you will the moment you start working with biological problems you will see that the size of the data, the size of the matrices become very challenging. So, you will have to, you may not even be able to load a data set unless you try to cast it as a sparse matrix. So, these are the challenges that you have to normally worry about. So, and then you have hypergraphs, which are nicer generalizations. So, in hypergraphs you will see that, so in a graph is that right, whereas in hypergraph you can have more than one node connected by an edge, more than two nodes connected by an edge, right. So, you have A, B, C, you have a hyper edge that connects these three nodes, which means that there is no way to just have a path, that there is no notion of a path between A and B now, there is a path called ABC. And can you see where this would be useful already from what we have seen today? Metabolic networks, right. Metabolic networks are naturally represented as hypergraphs, because you need A and B, you need glucose and ATP to produce glucose 6 phosphate, you can't just convert glucose to G6P. So, do read more about hypergraphs, they are quite important. And then we have the notion of a converse of a graph or a transpose of a graph and so on, and basically you invert the edges. If A is connected to B, you disconnect it, and whatever other edges do not exist in the graph you add it. So, essentially the sparsity will also invert. The graph is 3% sparse, the inverse will be 97% dense, not sparse, right. And you can have a complete graph or a clique, right, where every node is connected to every other node. We will look at these in little more detail in the next lecture. And you also have a notion of a walk from A to B, right. So, a walk is essentially node, edge, node, edge, node. So, A take the edge AB, B take the edge BC, reach C. So, it traverses nodes and edges. So, this is very important when you look at graph algorithms. We will not focus on graph algorithms in this course, but we will instead study how various network parameters, various network topologies are used to understand and infer about graphs. So, I think I will stop here for today. So, today we looked at a few more types of graphs and how one represents metabolic networks and what are the advantages and disadvantages or the kind of interpretations one can make with different types of metabolic network representations. And we also overviewed some concepts of sparse matrix algebra of storing sparse matrices on a computer. In the next video we will look at some more motivation for why we use graphs and the fundamentals of network biology beginning with various types of network parameters.